Yeah, thanks so much. I think you told everything about me. I don't even need to talk anymore. You know everything, including all my collaborations. I have long-standing collaborations in China, and I appreciate the cultural context. And I'm really happy to be here. And of course, as probably many speakers, I really admire and respect the work of Nikos. And he comes mainly from physiology, and I come maybe more from the psychology and the mind, but somehow you meet somewhere. And one key, I think, convergence could be the time scale. And I think it's often neglected. Philosophically speaking, the brain has its own inner time. Yeah, that goes an idea which goes back to Kant in the Critique of Pure Reason. So, and I take this inner time very seriously. As a good German, you take things seriously. And so I tell you a little bit about the time of the brain and its potential physiology and its potential relevance for mental features. And what do I mean by time? Just a very quick first approximation. By time here, time scales, I mean duration. Yeah, duration of processes. This is the archaeological time scale. You see, these are much longer time scales than our brain has. So these are durations of processes, like your heart has a time scale of 1 second. Usually your respiration has a longer time scale. And probably one of the unique features of the brain, of course one needs to be careful with the term unique here, is that it has a multiple layers of time scale, just like here the archaeological time scale. And I want to give you a little bit insight into these time scales. They're usually in the background of the brain. We usually just take them for granted. Even more, to my dismay, we often cancel and average them out in our data. So when you come to me with an experimental paradigm, a cognitive paradigm, and you say, Northoff, can we collaborate? So the first thing I will draw your attention is to the temporal structure of your paradigm. What is your intertrial interval? How's your stimulus duration? Because that, the brain encodes that in an ingenious way. So and the question is about the how. Yeah, so first I want to give in my first part of the talk, a brief, what are intrinsic neural timescales, very brief. Then I show you one potential key function, temporal integration and segregation of input. And you see, my talk will be much about the most basic layers of input processing. So I will not go into higher order cognitive elaborations, what happens in the brain and information integration and global neuronal workspace and all of that. I start much earlier, go to the very basics, the bottom, the input processing and how that is shaped by the timescales. Then I will show one line of research which particularly combines imaging and computational modeling, where we say that maybe recurrent connections, intra-regional recurrent connections. So there are various terms around will be key for intrinsic neural timescale and the autocorrelation window. And then I will show you in the last part some possible applications, role and function of the intrinsic neural timescale for brain-mind phenomena. I focus particularly on two phenomena, consciousness and schizophrenia. You see other applications on my website. So I come originally really from the opposite end, where probably you are coming from. I come from the mind, and then I try to make my way to the brain, and then I see, oops, I need to understand first the brain before I go to the mind. And it's amazing how much that even coming from the mind, I really need to get into new ways of analyzing the brain data. Yeah, so and that I think where the convergence point. So let me start again with another brief definitions. What are timescales? Timescales, the time allowed for or taken by a process or sequence of events. And there's yet another timescale which come more from the cognitive side, particularly from the group of Hasson et al. From Princeton, intrinsic neural timescale, the time window in which prior information from ongoing activity or stimulus-related activity can affect the processing of newly arriving information. So I will, here I marked this first one green because my focus in this section is on the green one. And in the next section on temporal integration, segregation of input, I will focus more on the second one. Very brief update, how can we measure the brain's intrinsic neural timescales to what is called the autocorrelation window. It's a standard method. Murray et al. 2014, Nature Neuroscience, he did this in monkeys. Honey et al. 2012 did this in intracranial recordings and has been done also in EEG, MEG, and more recently also in fMRI. And one key method is basically what you call the autocorrelation window. So basically you correlate the activity at t.0 with time lags at t1, t2, t3, t4, and so forth. And then you basically correlate the activity across all the time lags. And that gives you what is called the autocorrelation function. So this comes mainly from engineering and signal processing. And usually, traditionally, you say, okay, the degree of 0.5 correlation, that's where you calculate your autocorrelation window. But there are various measures. We are just, it's still in revision. We got some very good revised comment. We put in a whole software together for different ways of calculating intrinsic neural timescales. And I know that it's very small, it should be larger. So there are basically two main groups of calculating timescales. So one is model-free, here like the autocorrelation window, and one is model-based. It's a Bayesian way, actually from Zarate et al. 2022-23 introduced that in a paper in Nature Communication. And so that various method, and it really depends also a little bit on what your aim is. So we're just revising the software, and we also have a whole algorithm for which method, which question. So some of the basics then, the next question, how are the intrinsic neural timescales distributed in the brain? Obviously, they're not just there during task-evoked activity, as in the studies of Hasson, but they're already there in the resting state. So you have different durations of ongoing spontaneous processes. I think in the first way you can formulate it like this. And what you see, it's a hierarchy of intrinsic neural timescales. It really follows the topographic hierarchy from unimodal to transmodal cortex. So roughly here you can see this. This is an MEG study. Also, it has been done in fMRI too. Here, the blue ones are the sensory regions, the unimodal regions, also called the periphery. And the red ones are the transmodal regions, include the fault mode network, central executive network, and that's also called the core. And you can see that is really the intrinsic neural timescales. The autocorrelation window is much longer in the red regions, in the transmodal and the core regions, than in the sensory regions. So, and that's basically, there's a whole line of research now really where this unimodal transmodal topography is really supported by different gradients. Interestingly, there's also a gradient of neuronal excitation, that your neuronal excitation is higher in the transmodal than in the unimodal region. And also functional connectivity pattern, the McGill group and the Jülich Germany group is one other key players in this. And you see this also for different anatomical distinction of the core periphery, unimodal, transmodal. And what is important that these, let's say, temporal gradients really follow the spatial gradients. Yeah, so this is the, you probably know the ones who are into this gradient stuff, the paper by Margulis et al. 2016. So PNS, so we really followed exactly the same kind of pictures. You see a similar picture in his work, but here we elaborated it for our intrinsic neural timescales. And in his case, it was.
Sorry for interrupting, but it's important for me to understand that. Which signal time is it for that?
This is MEG.
Only MEG.
This is MEG.
Is the whole range of the signal, or you are taking different types? Like
this is no. This is 0.5 to 40 hertz. 0.5 to 40 hertz. That's a band pattern. So it's not specific frequency band. So yeah. So it would almost go, if you were calculating the ACW just in one limited frequency band, it would almost go against the nature of the timescales. Yeah. So this is really, we did this, but the results are problematic. Yeah, it's really a global temple feature. And of course, it's closely related to scale-free dynamics. However, when you calculate the power law exponent for scale-free dynamics, they have a correlation, but it's around 0.5, 0.6. So they're not the same. And we have various findings where the PLE, power law exponent, and the ACW dissociate from each other. Yeah, I don't show them here, but you can see this in some of our papers. So that really, let's say, that also really means for me that maybe the frequency domain, the scale-free dynamics across whole power spectrum, has a slightly differential role than the time scale. Yeah, we are not fully clear yet, but that's sort of really, we observe this dissociation again and again. So here I'm really on the temple side, I'm a global guy. Yeah, and I think when you look at the different specific contribution of the bands, yes, for very specific cognitions, but ultimately, let's say in psychiatry, not a single band is specific for a disorder. Yeah, and I think the recent emergence of the aperiodic nature of the signal also tells you the lesson you need to consider the whole spectrum. So here I'm really, you will hardly see any findings in a specific frequency band from me, because we simply don't get there. Yeah, because you really need to analyze the whole power spectrum. That's my deep belief. And the autocorrelation window too. Yeah, so here it's amazing, and this is probably for you no surprise. You really see a convergence between the temple and the spatial domain. Yeah, they really intimately linked. Let's say if you operate in a slower frequency domain, you're by default more spatially expanded. You involve more regions than if you operate in a faster frequency domain. Yeah, Buzaki already described that a lot. And that's really, we can find this in our data too. That means, let's say, if you have a cognitive task, if you operate the task in a very fast mode, you might have a more restricted involvement of different regions than if you operate in a very slow mode, you have by default a more spatially expanded mode of activation. Yeah, so these are basic principles. So first conclusion, very short, you can look for details on our website. And so for us, timescales are really an intrinsic feature of the brain. I know this is a strong statement I'm giving here, but you will see later, particularly in my brain-mind finding, when your timescales are changed, you really change the output and the function of the brain, and also the neuronal processing. So that's, and for us, timescales are really a most basic feature. That's why I say it's an intrinsic feature of the brain, and they shape both the topography, unimodal to transmodal, and the dynamics, shorter to longer duration and processing. And what you basically, in the sequel of the talk, is basically trying to an elaboration of this intrinsic nature of the timescale. Yeah. And that's what I'm going to the next one. So what on earth do these timescales do? So remember the two definitions. So now I focus more on the second definition. So what do these timescales do when you have to process inputs, let's say during task state? Yeah, so usually what we do with the task states, we average across trials, we take out the temporal fluctuations across trials. By that averaging process, we calculate some event-related potential or event-related activity, or the beta value in fMRI, or we do a time frequency, again, averaged across events. So we take out particularly the longer time scales in the background, which might shape because we basically average them out when just focusing on the short interval after stimulus onset. So here, now I show you a couple of studies showing that these timescales might be relevant for integration and segregation of input. So I usually compare, you will see this later, timescales with windows. So unfortunately, this room does not have a window. So usually I like to show the windows. Look, there's a window and you see the outer environment through the window. And if you have different window sizes in the room, you see the same outer environment in different ways through the different window sizes. Zoom in, zoom out. And that's the way you probably have to imagine the brain's timescales too. So here I show you various lines of support that maybe there's really temporal integration and segregation of input. So we come here from the psychological level. I first show you some psychological level, and then I show you some neuronal modeling level support for that. So question is, do the timescales mediate temporal integration segregation of external inputs during states? So here we conducted a psychological paradigm. So basically you have to associate, first look at A, you have to basically associate a shape, triangle, circle, or whatever with either you, yourself, or another person. It's a self paradigm, but that doesn't matter so much. Now here, important is that you basically have to associate, you learn, okay, in this session, the triangle should be associated with yourself and not with a friend or the stranger. And it's amazing, then you have really a higher accuracy, faster reaction time when you associate the triangle with your own self when compared to another person. So then you shade, okay, maybe now instead of a triangle, you associate the circle with your own self. And again, you have this higher accuracy and faster reaction time. So it's a self-prioritization or self-association task. It tells basically psychologically or psychodynamically put how narcissistic our brain is, because it's really amazing. It works. Whatever you associate yourself with, you process it faster and more accurate. It's amazing.
Possibility that it's simply easier to process the self because we know it very well. You present another stimulus to another person, the brain has to think about it a little.
Yes and no, because here the stimulus itself is not intrinsically self. You change it, yeah. But of course, I mean, we developed what we call a baseline model of self, because basically you process the input against a certain standard or reference or baseline. That's yourself, yeah. And that's in accordance with the fact that we see this rest-self overlap, because your spontaneous activity is a baseline for any kind of task-evoked activity, yeah. So the typical view of the self is it's a higher order function above any kind of input processing. It's the elaboration of the input. I would say, no, it's the other way. It's already the most basic input process, because you set the input against your spontaneous activity, and that is highly associated with the sense of. So we have a whole line of research, and that's probably what it is. Yeah. So, and it's really amazing. So you change once this, and in each case, you have associated with yourself, and you have a better processing. It's very consistent across domains, modalities. It's amazing. Yeah. When do you ever get something like that in psychology? Yeah. So please don't cite me on that one. Yeah. So, and then what we did, okay, we tried to do, okay, now I see we have a pointer here. You see that it works. That's this one. Okay, it doesn't matter. So here in the lower, here in the lower part. So then we said, okay, we build a delay in that, because we wanted to say the time scales of this effect. Yeah, on the psychological level, here you see here we present the triangle, but now the U is delayed. So we introduced some little delays in that. So basically test how much we can delay this self effect. And on the psychological level, we indeed observed that the self could really delay this effect even for 700 milliseconds. Yeah. So this is relatively long. This is in the delta frequency range, as you can see. So you can still have this self effect. That's so easy and so clear. Okay, so here's the, thank you very much. So here's the delay. Yeah, you see the delay, and here the association with yourself. So we disentangled the two. And then what we measured EEG, now we looked for EEG in the resting state. I show you some selected results from that. And here again, we coined out these cortical midline structures. So this is now EEG. And what you can see here, that basically the slope of the 4 delays on the psychological level, the slope to which you can delay your self effect, correlated directly with the autocorrelation window in these 2 cortical midline regions. Since we really tested here basically for temporal integration, because in order to solve this task, you need to integrate this one with this one. but we had the delay. So it explicitly required temporal integration between these two stimuli, and we could see the degree, the length of your autocorrelation window. The longer the autocorrelation window, the better you could cope with this delay and still maintain your self-prioritization effect.
That's a clarification question. So what is the subject?
So the subject is asked to do just to associate here what I explained here, associate yourself with the triangle. And another block of the task, you associate yourself with the circle and the triangle with the stranger.
They have to do this on it?
No, it's just self-associations. And then, of course, click the button. Yeah, and then they present, so that's what they learn for that block. I tell you, so now in this block, you associate yourself with a triangle. So, and then you need to, I present you various combinations and you need to click right or wrong. Next block, I reduce you to a circle and so forth. Yeah. So in it's really testing self-association. Yeah. How do you associate an external input with your own self, as distinguished associating the external input with a stranger or a friend? Of course, it's a very cognitive task. Yeah. But ultimately, I would argue it's basic input process. Yeah. And that's why we also correlated the resting state. Yeah. Because we said, okay, look, already in the resting state, you must have the proper time scale. That's what we measured with the ACW here. And say, okay, the longer your ACW in the resting state, the better you can overcome these delays. Yeah. And that an indirect support that this is really about temporal integration. Yeah. Here I present you another study, also again about the self. So we here presented a naturalistic stimulus, so you had to listen to your own story versus another person's story, 8 minutes. And of course, obviously, listening to a story, you need to integrate. Yeah, you need to integrate from sentence to sentence, from word to word, which admittedly we didn't measure explicitly here. And we again, the self differences are amazing. I must say, I'm doing the self stuff since 20 years. I'm amazed how different the self is. So now you can say I'm a highly narcissistic researcher, but it's really amazing. The self yields constant differences. It's not trivial. Yeah. And here we could see, for instance, here, while they were listening to the stories, they had an online where they had to shift their mouse about the emotional engagement. So basically, we saw in the movement of the mouse, we saw much more distance. They were moving to further parts of the screen in the self. So you're more spatially expanded than in the other. And the same was here with the velocity. You're faster, you're moving faster because we could analyze the mouse movements as the self versus the other, and the same here for the variability. So it's amazing. They're really processing advantages of yourself. Yeah. And then we also calculated again my beloved autocorrelation window. And what you can see in the resting state, you saw the shortest ICW, and then during the task, because you need to listen, you need to prolong your autocorrelation windows to temporally integrate. Yeah, and again, you see the self effect. And that's an effect, the prolongation of the autocorrelation window. You see in various paradigms, also other groups doing attention, working memory, they prolong the autocorrelation window, meaning, and that's very important, it's highly flexible. It's highly dynamic. You will see papers coming out now where we calculate also later the autocorrelation window in a dynamic way, sliding window. Yeah, so it's highly dynamic. Your time scale. So let's say if you imagine the windows here, the windows will constantly shape their size according to what happens there in the outer environment. Yeah, it's amazing how adaptive your brain is. I mean, this is for me one of the most amazing things on that. And there you see lower right, the respective topo map. So then here's another study. You see, we really try to get at this temporal integration segregation on a psychological level. So this is a meditation. So we have a strong collaboration with India in Bangalore, where we study now meditation. And it's unbelievable. If you've ever been there and what kind of meditations they have, it's unbelievable, and the philosophical background behind. So here we show you and we look particular for what exactly happens during the different meditation forms. So here, it's on the upper left is a shunya meditation, where you basically don't focus on anything. You just let your thought contents pass by, and you require wide attentional windows, meaning temple windows. Then on the right, you have what is called the Vipassana meditation. You focus on single body parts. So you scan, you focus on your nose, on your right ear, on your left ear, on your hands, and so forth. So you really need segregated attentional windows. Yeah, you need high degrees of temple segregation, and mantra chanting is in between. So now the hypothesis is maybe the duration of the temple windows on the level of attention, which is the higher or lower degrees of temple integration, is somewhat mirrored in what we call the autocorrelation window, in the temple windows on the brain. So here you see why we do this, because the assumption is maybe there are corresponding window duration on neuronal and psychological levels. So, and that exactly turned out to be the case. I should here do the mouse. I can draw your attention to this one. So here you see this is real data. This is real data. So you saw the shortest autocorrelation window in the Vipassana meditation, where you required clear temple segregation in your attention. Whereas in the Shunya meditation, where you had, this is EEG, by the way, where you had the longest temple attentional window, here you saw a much longer temple window or autocorrelation window also on the side of the brain. So that's sort of the idea that maybe there's a sharing of the time scale duration between the neuronal and the mental level. So that later we will speak of a common currency. So now the question is, you can say, Northoff, all good and fine, but you didn't really show me about that there is real temple integration and segregation. Yeah, it's only at best indirect, inferred from what happens on the psychological level. But you really want to show the temple integration segregation on the neuronal level. So this is now what we did. So here we did a simulation modeling. We use the Chaudhuri-Wang large-scale biophysical model, which is mainly based on monkeys. And so this is a biophysically realistic model, large scale with the proper regions. And what we did, so we stimulated this model, meaning V1, with inputs and followed how that input is processed in the subsequent regions in the model. Now, where is my mouse? Okay, now the mouse. Here it is. Okay. Okay. Yeah. So here you can see, and of course, no surprise, you know this much better than I do, that the temporal duration of your input stimulation is very well represented in V1. You see really here the duration of the visual V1 activity changes corresponds more or less exactly to the temporal duration of the input stimulation. So now, but when you now go from unimodal to transmodal regions, that becomes much more blurred. And when you go particularly to 24C, here's a red line, anterior cingulate cortex, you see that that activity is basically temporally smoothed, so that you cannot distinguish in the 24C, in the cingulate region, between the three different inputs and their duration. So there must be some kind of temporal integration going on. So here you have high degrees of temporal segregation. Here you have high degrees of temporal integration. And that goes very well with the unimodal transmodal topography, being autocorrelation window shorter in V1, you can see this here, and longer in the 24C. So then you say maybe that does not hold for longer inputs. So then we did the same, we stimulated the system with longer inputs, and we saw the same pattern. Yeah, there's a temporal smoothing of your activity duration in the 24, and that can only be due to the longer time scale / autocorrelation window in that region. So you really and that, of course, I don't need to tell you that what that means for cognitive processing, what it does to the input. And then the one who did the simulation, the models, the Yazir Chatal, he came up with a very nice idea. There is in the Chaudhuri model, there's also a hierarchy index. There's a little H. When you look into the details, there's an H in the formalisms. I didn't know that either. So he came up with it. I said, okay, now we play around with that hierarchy index. So meaning maybe the timescales differences disappear when you set this hierarchy index to 0. And I think it was a really ingenious idea because you see the result. So here you see for each region, you see a different hierarchy index. And you see the typical pattern with the temporal integration, temporal smoothing of the signal in the 24C. But now when you set this hierarchy index to 0, yeah, all regions the same. So you don't contribute a different hierarchy index for the different region. So suddenly you see very nice, the same signal duration in all regions. So here it's really a topographic and whole brain global effect. Yeah, what I find really amazing, because you calculate your timescales intra-regionally, but it's a global local topographic effect. I think that really suggests your global topographic structure is absolutely key. Yeah.
You think this change in hierarchy can be accomplished with consciousness versus non-consciousness?
I will show you data later on that. Yeah, you're reading my mind. I must be careful here. Okay, yeah, so that is, I found that quite amazing. So how does the whole thing works? I show you. So this is a schema, but I think it's implied, and we have another paper now in revision where we show exactly that. So, so you present different inputs here, and let's say you have medium length of your autocorrelation window, and here you see the objective duration of the input. And let's say if your autocorrelation window, let's say in V1, is long enough to cover these two inputs, then you put them probably together in one evoked activity. Meaning in your evoked activity, you do not separately represent these two inputs anymore. And let's say now, however, if you have a very long autocorrelation window, and you will see that later when I present the data on the disorders of consciousness, then your autocorrelation windows become very long, meaning you put all those inputs together and don't make a difference anymore, and so you have one evoked activity. Meaning here, you cannot distinguish between these two inputs anymore. So, and so this is really a key thing for how much your input processing might be already shaped by your autocorrelation window and the subsequently evoked activity. Here in this paper, we really get into the details of the relationship between the ACW and the evoked activity. So I hope I could make a case for that, though tentative, that this time scale may be key for temporal integration segregation of input. So now the question is, what is the potential physiological mechanism? So, of course, there could be a variety of physiological mechanisms, but here we focus on recurrent connections, and I present you some various lines of evidence for that. And when I speak of recurrent connections, I do not mean interregionally, I mean intra-regional. So within the same regions, the excitatory, for instance, excitatory populations refer to themselves and have basically recurrent loops. You have different forms in the Wilson-Cohen model. You distinguish different forms of recurrent connections: excitatory, 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 inhibitory, 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 excitatory. I mainly focus here on the excitatory, excitatory. So, and I show you one study where we again this Yazir Chatal, the one who also has a hierarchy index, so he does all the modeling, and so he first did some animal data, mice data, then human data, and then the modeling. And I will develop that a little bit so that you get an idea what we are talking. So the first thing, he took up an online data set where they did calcium imaging in mice. and no surprise what he observed. And they were freely moving around, and then they had to do various kinds of tasks. And that's what you can see here, locomotion, rest. And you can see during the sustained resting periods and during the locomotion, they really had a much longer ACW. So this is basically what I already showed you. When you perform a specific task, your autocorrelation becomes longer. Yeah, each region here, each dot is one region. And you can see here, this also holds for each of the mice. These were 5 mice in total. And for each of the mice, we could really see this prolongation of the autocorrelation window during the locomotion compared to the resting state. And then he said, maybe there's a relationship between the resting state and the active state. So why will you do this? Because for me, the spontaneous activity, and you mentioned that already in my bio, that I really early on said, okay, what on earth is the role of this spontaneous activity? Everybody talked about it, resting state, but nobody really knows it. Maybe you guys know this. And now, I think since 4 or 5 years now, finally they found a solution, say, okay, resting state is internally oriented cognition, and task state is externally oriented cognition. However, I don't think that, let's say, internally oriented, like self-referential processing, episodic memory, and so forth. And however, I do think that your spontaneous activity really serves as a baseline, as a default. And as far as I understand Reckle, that was also his original idea in his 2001 paper. I can get more into that. So now we say that's why you see in our papers a lot of rest task correlation, rest task prediction analysis. Because we say that your task evoked activity stands relative to the resting state. And that indeed, we had a lot of findings on that. So here we did the same. And he was particularly interested in saying that maybe your dynamic repertoire of the resting state's ACW may have an impact on its degree of change during the locomotion, during the task. So he calculated the variability of the ACW. He took 10-second windows, non-overlapping 10-second windows, 10-second windows, and calculated the variability of the ACW from the resting state, and then could observe a negative correlation with the rest task mean change. So that's what you can see here in the upper right, the negative correlation. You can see this here, meaning the higher my variability in the resting state, the lower of the ACW, the lower the change of the ACW from rest to task. And again, you could see that hold for all of the 5 mice. So it really seems to be a robust finding. Then he did the same in a human data set. So here, this is the same data set I presented you before with the listening to the own story. I remember there were 3 conditions: a resting state, listening to the own story, and listening to another person's story. But now he analyzed that same thing, 10-second periods of ACW in the resting state, variability SD of ACW in the resting state, and then see how that predicts the rest task mean change. And you can see that here already, as in the mice data, in the both conditions, you have a higher, longer ACW. I showed that before, but now this slightly different calculated across all channels also. And here you can see the same negative correlation. So meaning higher variability in the ACW relates to lower rest task ACW mean change. So what could that mean? That if you have a higher dynamic repertoire in your resting state, more variability patterns, you're less perturbed by the external task. Yeah, because ultimately your brain wants to keep its time scale stable, which of course is an assumption. So then the next question said, okay, where and how, what is the potential physiological basis of this relationship of the resting state ACW variability with the change during rest task. So for that, he calculated a Wilson-Cohen model, neural mass model with recurrent connection. As I said, when I speak of recurrent connection, I mean intra-regional recurrent connections. And first, what he showed, you could really replicate the human and the mice findings in the model. So you saw again, we did some simple stimulation in the model, and you could really see a difference in the ACW, and you could also see the negative correlation. So that basically said, okay, our model must be somewhat valid. And now we said, okay, now we can test the, really test how this rest ACW and its negative or relation to the ACW rest task change is related to the recurrent connections. So he varied now the different degrees of intra-regional recurrent connection and tested how that affects your rest and task ACW. And you could see here, let's say, if he keeps the recurrent connection on a very low level, on a non-physiological level, there's no difference between rest and task. Now he increases the recurrent connection to more physiological levels, and you can really see you have a clear difference between rest and ACW task. However, and that is really interesting, there's a certain ceiling effect, meaning when they're too high, now they're no longer different anymore. And when you make them even higher, the recurrent connections, suddenly the opposite happens. So, and even higher. So together, this amounts to what can be is this kind of curve. So you have a certain average degree of recurrent connection, which seems to be optimal for maximal rest task ACW difference. Yeah. And I think I don't need to tell you, this is a basic, probably these inverted U-shaped curves are a basic feature of dynamic systems. We had a whole paper on that in 2019, coined average is good, extremes are bad. Yeah. So now let's say if we could develop this further, this could, for instance, serve for psychiatric. You know, I'm also a psychiatrist. For, well, where is my cursor now here? Okay, let me get that here. So let's say if we really put this on an individual level, the idea is maybe later we can really have a readout, let's say, for instance, where psychiatric disturbances are, like depression, I would assume they are more here. They have a too high degree of recurrent connection. I have this based on various findings we have. So you could ideally locate the individual subject and make prediction. That's sort of one of the main reasons why we do this modeling, besides understanding the basic physiological features here. Yeah. So, and then another line of support for the recurrent connections come from another thing. This is another Khan Keskin, also from Izmir, actually. And so we, as you know, we did a lot of research on the self, on the temporal features, but also on the spatial features. So in a large-scale meta-analysis of all kinds of self-studies, we basically established this three-layer topographic model of self. So all regions associated with interoceptive processing of the self are here, typically insular, dorsal anterior cingulate. It's a lot of science, parahippocampus. It's a lot of science network regions really associated with interoceptive features of the self. Then exteroceptive features, so these are typical tasks where you're like proprioceptive, or some of you may know him, cross-modal integration, Olaf Blanke. It's basically those regions. This is bilateral temporal parietal junction, again the insula and premotor cortex, and then mental self-processing is a kind of task I presented you that's probably associated maybe with the mental self-region, cortical midline structures, the fault mode network. On a side note, one amazing thing, this is a nested hierarchy. So the regions which are activated here are also recapitulated here in the activation, completely independent studies. So it's really a nested hierarchy. Clinically, this makes perfect sense, because even if I have mental problems, I always say always occur against some somatic experience, like in depression or anxiety. Yeah, so this nested hierarchy, and we really now develop that, apply it to different studies, use it for differential diagnostic. So let me just drink some water in between. So, but here we said, okay, maybe this serves to distinguish self and non-self regions in the brain. So regions which are more important for the self and less important. So see, it's all about, for me, the intrinsic topographies of the brain, because that's what because I'm interested after these intrinsic topographic features, because I say they are key for mental features. I will show that to you in the next part. So here, and the first thing, so Kahn came up with the idea. I loved the idea. So he said, okay, I just want to see whether self and non-self regions, so the self regions or these regions and non-self regions have slightly different dynamic and topographic features. Yeah, because that means if there is an ingrained or intrinsic self-non-self difference in the brain, which is key. So, and he computed the myelin. There's also a unimodal transmodal gradient for the myelin known, and the ACW, and he also calculated the global activity, global signal correlation. This is basically a global functional connectivity, and you can calculate that in different ways. So, and what he first found, no surprise here, the myelin was lower in the non-self regions than in the self regions. ACW was longer in the self regions than in the non-self, and GS score was higher in the non-self regions. So this is no surprise, expected. Then we also, he did various correlation and also Bayesian statistics. How are these three features related with each other? So first, of course, no surprise, the higher my myelin, the shorter my ACW, and then the opposite, the higher my myelin, the higher my GS score, because that goes with the unimodal transmodal distribution. And then he calculated mediation models, and that's basically the relation he got from the myelin as an independent, GS score, global functional connectivity, as a dependent, mediated by the timescales, yeah, by the ACW. So that tells you that's really important that your timescale operate a temporally structural impact even on your global signal. So the next thing said, maybe that is related to recurrent connection. So again, Wilson-Cohen model, and particular focus on excitatory recurrent connection, as I said, intraregional, and the basal tone neural excitation. And first, I spare you some of the details. He did also single node simulation before he entered the whole model. You can read the paper. It's a quite really thorough analysis. And here I directly want to, because these are the empirical results from the fMRI HCP 3 Tesla data set. And you can see, so this is a myelin. And here also he distinguished between transmodal. He compared the self-non-self with the unimodal and transmodal. Because the question is you could raise, is the self-non-self distinction just the same as the unimodal transmodal? Or is it slightly different? Do they overlap? That's it. So he distinguished between transmodal self regions. Let's see that here. Transmodal non-self regions, unimodal self regions, unimodal non-self regions. And what he could see that in both cases, the transmodal and the unimodal self region differed from the transmodal non-self and unimodal self. So that means the distinction between self and non-self is not a trivial one. Yeah. So it is probably a nested topography within the unimodal transmodal topography. That's what these data support, because you really see differences in myelin, in ACW, and GS core. So then now I show you the simulation data. So, and what did he do in the simulation? So you can now test the relationship between the ACW and the recurrent connections and the excitation by varying the degree of recurrence or excitation. So the control condition is you fix them. Yeah, you do not let the recurrence and the fixation, the excitation values adapt to the parameters of the model. You just fix them in the model, and then you calculate the ICW in the model. And that's what he did here. And you could see none of the topographic differences come out when you fix the recurrence and fix the excitation. So that indirectly suggests excitation recurrent might play a role. So then he said, okay, I start with the excitation, I vary that, and I let the excitation degree in the model adapt to the data and fix the recurrence. You see somewhat a curve which somehow resembles this one, but the best one you get when you vary both, when you no longer fix both the recurrence and the excitation. So, and you see this curve is remarkably similar to this one. So, you know, modeling is not an easy one. So you're really happy when you see that. So, and then he did the same for the GS core, for the global functional connectivity. Similar thing, you fix the recurrent, you fix the excitation, no differences at all. You fix the recurrence, you let the topographic excitation free, somewhat, not really, and the best one when you have both varied. So that strongly suggests that recurrent connection, intra-regional excitatory, and the basal tone of excitation might be a key role in modulating the ACW and also GS core. We also did the global coupling strength in the model, which was much better related with the GS core, of course, no surprise. So what does this mean? So this is, of course, an interpretation, but still. So we call here on the basic level, you have the excitatory recurrent connection, you have the basal tone of excitation. That shapes particularly your timescales, your ACW, which in turn provides the transition from myelin to global functional connectivity. And what does this now mean on the psychological level? We haven't shown that yet. But of course, what you mean, you have more, if you have a longer ACW, you have more recurrent excitatory connections within your regions. Of course, you process the stimulus in a longer duration. Yeah? So that means you process the stimulus longer, and that longer duration of the processing might yield your self-reference. So the longer I process the stimulus, you might have the feeling, okay, that belongs to me. And if it's processed only short, then you say, okay, has nothing to do with me. Of course, that's something now to be tested in future studies, and that's exactly what we're doing now. So conceptually, it means that maybe your neuronal recurrence on this level might be mirrored in corresponding levels of psychological recurrence. And where it puts a smile on my face, that maybe what we call self and assume all kinds of higher order special cognitive mechanism is just a feature of the duration of the temporal processing of your input. Yeah. So here you see a probably temporal shaping of your presumably higher order cognitive feature. So this is later at the end of my talk, what I call spatial temporal neuroscience, and always puts a smile on my face. Basically deconstruct the higher order cognitive assumption and all these special entities they assume just by basic spatial and temporal features. So after this, now I come to some of the applications. First, I want to show you some studies on INT and consciousness. So we did this in different states of consciousness, in sleep, ketamine, anesthesia, sevoflurane, and also a coma, unresponsive wakefulness. And what you can clearly see, almost parametric here, in 1 and 2 and 3 REM, you see a prolongation of your ACW. Yeah, and that's also here in the anesthetic state, ketamine not as much, sevoflurane much coma very much. So that really means that the input processing is blurred. Yeah, they integrate all the inputs, cannot segregate them. Yeah. Then we said, okay, because alpha is so much important for input processing, maybe relate the ACW to alpha again in patients with coma, and that's what we did. So here we developed different measures of the alpha peak. This is all EEG. We did and broadband. This is of course only alpha. And you see here, we show you different types of patients. This is a healthy subject, good alpha peak. Some patients have a weaker alpha peak. In some patients, very interesting, the alpha peak shifts to the theta peak. It's probably part of the slowing of the power spectrum. In many patients, you don't have a peak at all. So this is also statistics. We had different measures of the alpha peak. I spare you the details. Developed these different measures of the alpha peak, and they were all highly different between the coma patients and the concept. Then same patients is a different data set. We replicated the prolongation of the ACW, calculated the ACW here in different ways. I spare you the details, but what is important that we then linked the ACW to the alpha power, and we could really see that if you have longer ACW, you have lower the alpha power diminishes, your alpha peak diminishes. So meaning your temporal integration, your long autocorrelation window, calculated from 0.5 to 40 Hertz, operate in the background and impact your alpha peak. If these windows are too long, on the longer time scale of the broad frequency range, it affects the power in the very specific shorter time scale of the alpha range. Yeah, so this is basically a background foreground thing here on the neuronal level. And interestingly, that correlated also with the level of consciousness. You see down here, it's a mediation model. You see the alpha power as an independent variable, and you see the ACW as a background mediator, and then here's the level of consciousness. So again, here could be a potential diagnostic marker, yeah, on the clinical side. On the basic side, you really see that the ACW in its broadband frequency range seems to operate in the background, has a temporally structuring effect, how the alpha power affects your level of consciousness. Yeah. Here we have another study. This is now fMRI, and we can see the same thing. This is now fMRI in the sleep state, and in the sleep state we had a resting state, awake. We had a sleep resting state, and we had a sleep stimulus state. They were presented with an auditory movie, and from which they did not wake up. This is, of course, the question the reviewers raised. I know it's hard to see, but you can see here, this is the awake state. This is the sleep state, sleep resting state. You see the ACW is longer here when you look at the details than here. So as I said, this is now fMRI, so we're in the infraflow frequency range. And interestingly, during the stimulus state, in the sleep state, the movie, the ACW became shorter again. So you can see this here. These are basically plotted. This is the sleep. This is the sleep. This is the awake resting state. This is a sleep resting state, and this is a sleep task state. So that suggests that maybe your ACW, the reactivity is not tied to consciousness. It's a basic processing feature which is not tied to consciousness. It's reactivity to stimuli. So then we also did, just to get a little bit better idea what the ACW is about, the median frequency. We calculated the slow-fast balance, frequency balance in the power spectrum, which is called the median frequency. And no surprise, the power spectrum became slower during rest sleep, and then during the task it became a little faster. You can see here the opposite. So, and then we also here, sorry, in the next one, that's here. So then in the next one, we also correlated the median frequency with the autocorrelation window, and no surprise, the longer your autocorrelation window, the stronger the slow frequency power. So here you see a direct relationship between a time domain and frequency domain, and what probably drives the longer ACW. So then we also did another biophysical modeling here, because we said, is this effect of the ACW, the shortening and lengthening, what is the physiology of that? So here we, the first author of this paper came up with a clearer idea. We used, again, this Chaudhuri model here from 2015. We said, okay, how can we model sleep in the simulation? Maybe we just prolong the excitatory, we change the excitatory time constant. So instead of 20 milliseconds, we put in 60 milliseconds. And that's basically simulation of sleep in the model. And then we said, okay, if that is the case, maybe the ACW becomes longer if you have a longer excitatory time constant. And then how do you modulate the task? Yes, okay, you present different sine waves to the model. So, and that's exactly what you see. And what you see here, I know it's a horrible figure. I know we should have done this much better, but it is. So here you see the different brain regions in the model, V1, V2, V3, and so forth, up to the hierarchy, 24C. And you see the three different states. So you see the excitatory time constant 20 milliseconds, which is more physiological. Then you see a longer time constant in the middle. And then you see the additional sine wave stimulation, which is here. So you see the short time constant, long time constant plus stimulation with the sine wave. So that basically was supposed to model this is the awake state, the short time constant. The longer time constant is supposed to model the sleep state. And the longer time constant plus the sine wave is supposed to model the sleep state plus the movie. And the ACW results were exactly like that, what we saw in the empirical data. So we show a much longer ACW. Now this here on the y-axis is the ACW values in the model. You see a much longer ACW in the longer time constant. So that tells you also that the timing of your excitation might be also an impacting factor physiologically of your ACW. And then interestingly, the stimulation reversed that prolongation. You see that here in V1 as becoming much shorter again. The ICW, you see that here. And interestingly then, in the higher order regions, you didn't see the shortening of the ACW. So that really suggests that first, ACW plays a key role in consciousness in both fMRI, EEG, both in the infraflow and faster frequency domain. And second, that recurrent connection and the level of neuronal excitation, in particular the time constant, might be a key in that. So you can really hear the physiological features. So now, last slide on the consciousness stuff. And then I will briefly go into schizophrenia, 2 slides, and then finish. How do we imagine that on the mental side? So you say, yeah, Northoff, you have all the physiological features now. How does it impact your actual consciousness? So this is something, what we imagine that this is a schematic diagram, but this is based on our ACW in EEG data. So if you have a shot, you need a variety of different time scales. Here, this is a police and a thief, and they speak with each other. You need short time scales to pick up each word. You need longer time scales to make connection with between the words, and you need even longer time scale to make the connection between the two people, because these are all different timings. So since I'm doing all this timing stuff, I'm much more aware of the importance of timing in my daily private life, not only coming always too late, but also in things occurring, let's say, when you're in Teams or in Skype or Zoom or whatever, then suddenly there's this, the other one is slightly delayed. And if that happens more often, you're really confused. Yeah. So timing is unbelievably important. Of course, no wonder that I say that. So now when your time scale is longer, meaning you miss the shorter time scale, your input processing is more imprecise. So it's more blurred, your perception, and even not, and then it's completely blank. So I imagine the consciousness or absence of consciousness by these people to be just like a dark hole. Yeah, and we are trying now, of course, it's hard to prove this experimentally, but that's what we're trying now to do, also particularly in studies with meditators, that we get this phenomenological dimension clear. So I show you briefly an example from the mental disorders, schizophrenia. So interestingly here, we can really see in schizophrenia sample, and it's replicated in another sample, we see longer time scales in schizophrenia. You can see this here in the resting state considerably longer. And interestingly, what is also a very consistent finding in schizophrenia by others and ourselves, that the phase coherence, your intertrial phase coherence is imprecise. So what is intertrial phase coherence? You present a rhythmic stimulus like an oddball paradigm, and you see your phase is usually coheres nice with the timing of the stimulus. So all the phase angles go into the same direction at stimulus onset. And in schizophrenia, and that requires temporal precision. Yeah, all the Lakatos stuff, he really showed all that, developed that. So that your phase coherence is really temporally precise in the millisecond rate. Yeah, it's not only gamma, it's a steady state paradigms, but it's also alpha and theta. And now we also showed in the infraslow range of fMRI. So your phase coherence gives you with temporal precision. And here we can see that temporal precision is lost in schizophrenia. They have abnormal phase cycle variation. And for instance, one finding is here that you see here the phase coherence for the healthy subjects. Here, there's an oddball paradigm, 1 second intertrial distance, and you see the phase coherence is much lower in the schizophrenia. So that led us, these and various other findings led us to develop a temporal imprecision phase-based model of schizophrenia. So then we correlated the ACW with the ITPC, and you could see here the black lines are the healthy subjects, and the red lines are the schizos, and you see clearly that longer temporal windows increase temporal integration. This blurs temporal imprecision, and you have lower intertrial phase coherence. So again, you see the ACW somewhat operating in the background for a more longer, shorter timescale measure like the ITPC. So that's why we suggest now maybe it's the temporal layers in your brain. And that is no longer in schizophrenia. So that sort of temporal structuring is no longer occurs in schizophrenia. So maybe they have a dissociation between different timescales, shorter and longer. So now you say, how is all that related to the symptoms? I show you one example here. So in schizophrenia, it's often assumed that they have a basic self-disturbance. They cannot distinguish between self and environment. There's an inner and outer confusion. Typical example, delusions and hallucinations. And so how can we test that? So here we had a typical temporal integration paradigm, morphing phases 10 to 15 seconds, morphing phases from your own phase to another person's phase or the other, and you need to click when your own phase is over in another person phase. So that really requires temporal integration, because if you integrate all the stimuli too much, you will also see your own self when the other person's self is already there. So that's temporal integration on the paradigm side. Do this morphing stuff, and then of course we calculated my beloved ACW. Again, different data set. We could see the abnormal prolongation of the ACW here on the right in the schizophrenia, like in the other study. So it's a replication which doesn't happen that often in psychiatric literature. So you're always happy when you can replicate. And then we calculated a moderation model. We also measured the self-disturbance as a psychological, phenomenological instruments for that. This was done in collaboration with Denmark. And okay, now I need to find this. So what we observed, that your basic self-disorder measured in this psychological way, really this is the schizophrenic symptoms, general negative symptoms called, really was moderated by the prolonged intrinsic neural timescales. So meaning again, your timescales seem to somewhat operate in the background, and shaping the more event-related methods like intertrial phase coherence, or also event-related potential. This is now in revision, that paper. And then also some of your psychological features. Yeah. So that is really here. So I come to the end, to the conclusion, why I suppose the timescales are so important. Because for me, they're really key in your alignment to the environment. So here, this is a schematic thing. So usually we are confronted with a variety of timescales in our environment. I speak fast, I speak slow, I move, you need to follow this, you need to follow this, you need to follow the slides. So, and that probably your brain matches those timescales with its own inner timescales. Yeah, it's a matching, and we call this mechanism, and we're really trying to get into this mechanism, temporal spatial alignment. So here I give you an example. So you have a music, paradigmatic examples of different timescales. Yeah. Now, if here had a robot here, yeah. If the robot, now here it is. Okay. If the robot has only shorter timescales, no longer, it will only pick up the faster part of the music, meaning the robot will dance too fast. Now, if the robot has only the longer timescales, but not a shorter one, it will only pick up the longer timescales of the music, but not the shorter one. So you need a variety of different timescales. Ideally, that's why we get into the dynamic repertoire, the variability of timescales in the resting state, to optimally be adaptive and aligned to your environmental context. And sad but true, these examples are extremes, but you see this in psychiatry. You see the depressed patient, the dance is too slow. When you see them in music therapy, they're literally too slow. What happens in the brain? They're too slow. We have a whole line of research where we show that depression is not just a mood disorder, it's a speed disorder. These guys are too slow. And in mania, it's the opposite. Yeah, you're too fast. Literally true. So the other thing that, of course, now that's an inference that we say, the timescales, the repertoire of timescales here, this is a schematism, the species degree of timescales, this repertoire of timescales, make it possible for them to match or not match with corresponding timescales in the environment. So on a philosophical level, I would interpret the famous "What is it like to be a bat?" by Thomas Nagel, basically just a timescale. The bat has ultrasonic timescales, which we as humans don't have. So the bat can connect to ultrasonic timescales in the environment, which we cannot. So that's why it would be very interesting to a cross-species investigation of timescale, and also present them either with the same timescale range in the input or with different species-specific timescale range, and then see how they react. That could be done. So I already said that this room doesn't have any windows, but I show at least windows here in my slide. You can see this is the way I imagine to be. You have a whole window, you see the whole scene. Then you see subsets, you only see the leaves, but you don't see even that is a tree, or let alone that there's some mountains in the background. Yeah, and that's the way I imagine our brain to be, like a multitude of timescales which provide our window with the environment, with corresponding timescales in the environment. Why is that important? Here, present you a summary picture of empirical findings. So this is all based on findings that if your timescales are too long, your dynamics is too slow, and you almost focus always on the present. That's what you see in extreme case in depression. Now, if your timescales are too short, yeah, like here, and your frequencies are too fast, you always focus on the external environment, you suffer from mania. That's findings based. Now, if you have an optimal balance between longer and shorter timescales, and faster and slower frequency, you don't perceive the past and future anymore. It's distinct from the present. These are the kind of states people report in meditation and psychedelics. Yeah, so here you see that's, of course, and now we are really trying to test this here, particularly the middle state, together in collaboration with the Indian people, and indeed results support them. So second to last slide, the assumption is that's an inference now, and that's basically our overall working program. Timescales are shared by brain and minds. They're shared feature. So meaning I'm against invoking specific mechanism like information integration, global neuronal workspace, higher order for making consciousness possible. I rather go back to the basic intrinsic features of the brain and its input processing. The timescales, because I say that maybe timescales are shared on the neuronal and mental level. If our brain has only longer timescales, we also experience only longer timescales in our consciousness. We call that concurrency model, and we're really developing that. And there's, of course, not a one-to-one correspondence between the timescales in your consciousness and the timescales in your brain, because the timescales of your consciousness are very limited. 30, 50 milliseconds to 3 to 5 seconds. This is nothing. The brain has a much wider range of timescale. So that is impossible that you have a one-to-one correspondence. That's why we speak of a complex correspondence. And I hope I could convey my main message. If you want to read more, it's a recent textbook last year about that. Thank you very much.
Thank you very much. It's brilliant talk, inspiring. Do you have any questions? Yeah.
Yeah, beautiful talk. So I was wondering with regards to, so when we do monkey research, most of us here are doing, the time scales that our tasks are dealing with are very short. Now, I would assume that's why I plan to work on more complex tasks personally, where I can actually do additional time scale. We can talk about that further. But so in patients in a clinical setting, when you actually want to diagnose, or you want to use time scales as a marker, and how they integrate information through time, what is the reasonable time scale that you can actually play with? And what is the sufficient time scale to work with to be able to tell?
I can give you a short and a longer answer. Short answer, we simply don't know. I would like to know the timescale range of the brain. Yeah, I don't know. Yeah, I'm looking for that. I cannot find that information. Some people say what I'm doing is trivial. Of course there's time, yeah, but they mean the time perception. They don't mean this inner time. And for patients, it's a trivial. So we use the timescale thing, as you said, that's exactly one of my directions for diagnosis and also for therapy. So, for instance, in anxiety. So for me, anxiety is the disorder of too fast speed and irregularity. Ask the patients, that's what they will tell you. Not all this cognitive stuff and what you see in the little, oh, please forgive me. Yeah, that's what you see in the brain. Yeah, you see increased entropy, increased Lambert-Wave complexity. This is in revision. Yeah, in the brain of these guys. And even more interesting, you see us even in the breathing pattern. Yeah, you see increased entropy, increased Lambert-Wave complexity in the breathing, predicting your anxiety. See an anxiety like this. Yeah, and heart rate too. So now what do we do here? So what is the time scale I need to operate? So now what we developed is an individualized breathing therapy so that we titrate the optimal breathing rate flow, which reduces your anxiety and your thought racing. You know, they have this thought racing. They always coupled together. Incredible. Yeah. So, and for every subject, it's different. So when you look into the internet, slow breathing, standard rate, 6 breaths per minute. From our 50 subjects, only 10% had 6 as their preferred breathing rate. The other one had different. That's where the inter-individual difference comes. Because what the default is for you might already cause anxiety in me. That's a real problem, anxiety. That's why we do this personalization. And it worked. We see immediate effects on the anxiety. So even the timescale range, the functional timescale range, might be different between you and me because we grew up in different contexts. The physical timescale range, of course, is similar because you and I assume, share the same brain. Yeah. However, that physical timescale range might be different between the monkey and us. And I would love to conduct such a cross-species studies, test them with, measure their timescales, the variability of their timescales, their dynamic repertoire, and then test them with paradigms which have slightly different timescale involved in the time to make this matching alignment test for that. Yeah. And ideally, I would love to combine that with some dynamic analysis of the fluctuations of your GABA and glutamate. Yeah, the dynamic analysis, because that I assume somehow maybe those time scales might also correspond. Because you deal with a lot of methodological problems here because you have different sampling rates for the different levels. And trust me, it's a nightmare. I know from the behavioral stuff. Sorry.
Thank you, Dr. A short question. With such a repertoire of timing integration parameters, for example, would you have thought that there's some brain version, some amount of brain versions that mediate these kind of positions, like pacemaker? Or do you think the timing does happen simultaneously?
Yeah, very good question. So when I started all this, I had real problems that there are specific pacemakers in the brain. I can't really answer that question of oscillator in the brain, yeah? Because I'm not too much versed in this oscillator thing, but in the relations between these time scales and the oscillators, let's say for the different frequencies or pacemaker, one is more local, yeah, local tempo in specific frequency ranges. This one is more cross frequency. Now, there is an earlier paper by a Finnish guy in Busaki, 2003, where they assume a numerical algorithm between the different frequencies. So you have an optimal distance of 1 when you put this into an algorithm between the center of each of the frequency bands. So between, let's say, 10 Hertz alpha, 5 Hertz theta, I spare you the details. You have a maximal distance between the different frequency bands. And that accounts basically for their functional specialization, so that they don't mix with each other. However, so that's a scale-specific architecture. However, as we all know, we also have a scale-free dynamic architecture with the nesting of the different frequencies. And what I mean by time scale probably reflects more the latter. Yeah, the scale-free aspects of the time scales. But my point is that within this scale-free organization, the sequence of Russian dolls, you have specific time scales embedded. So meaning scale-free dynamics and scale specificity do not contradict each other. So what I try to do, that's now what we're trying to do. Let's say when I said the ACW operates in the background, operates on a longer time scale, and it shapes the shorter time scales in the more foreground. Like, remember the intertrial phase coherence, which was more scale specific. So I would argue there's probably a layer-like nested transitioning between scale-free and scale-specific aspect. Yeah, and I think that's also what we experience on our level of consciousness. I'm not sure whether it answers your question, but at least I get some kind of. Yeah, so it's a complicated. Yeah, what is clear, I do not believe that just the brain is a set of oscillators and then there's some secondary integration. That's definitely not the case. Yeah, okay, yeah, without really being able to give any argument for that, but I don't imagine the brain to be like that. Yeah, and that has huge consequences. You always have a global temporal background. Yeah, in your vision, you have a global to local differentiation. You first see the thing. Yeah, you first see the global thing or the global difference, and then you subsequently you'll be more able to specify things. The global to local differentiation, that I believe here. The foreground background model, from longer to shorter time scales. Yeah. So, and we have relations when we calculate our ACW with event-related potentials or intertrial phase coherence. It's usually a correlation about 0.3, 0.5, 0.4. So somewhat is a relation, but not really. And probably our statistical tools are not sufficient to account for that nested relationship. That's why we now look for measures of nestedness. Yeah, which I think is a key principle in the brain.
Thanks so much. I'm going to repeat partly the question that I asked. The signals, the signal that you measure very well is a multiple parts. Yeah. Will it be worth actually for that very interesting about going to base and understanding what exactly is happening to the frequencies?
That's why I'm here. Nicole, remember what I asked you. That's what I'm here. I'm missing the physiology. Yeah, you see how desperately I try to get through that through the modeling, but I'm missing the physiology and the biochemistry. And I know I need to get there, because only when I get those principles, then I can explain the mental stuff. I'm a deep believer. You need to make this detour. Remember, with this recurrent connection, I mean, in the self region, when I saw that you have more recurrent connection in the self regions. So what happens with the input? It's processed longer, and now we can test it on a psychological level. Yeah, so yes.
Very, very interesting to see what the data, particularly because some of the compounds are okay.
Yeah.
With each other.
Yeah.
So very interesting.
That's why I'm also interested in the in your biochemical stuff. Yeah, because ultimately we need to see the excitation inhibition fluctuating.
A very simple question you may have already checked. You see differences in ages.
Yeah. So it is known, let's say, that neuronal variability and ACW change in age. The ACW becomes a little longer. And actually, I can see this myself. It's very funny. So initially, then I couldn't remember certain things, but then I remember them later.
Yeah.
So and now I know. It's not that I'm getting crazy. Yeah, maybe I'm crazy anyway, but I remember them, but later. So you have a shift. I probably don't have as much faster frequency power anymore and shorter time scales, but the longer are still there, and I can still rely on them. And now, actually, on the other end, I just in Beijing, there's a student now who's really interested in the development of the self from baby on. And in the babies, you have early on you have relatively long INT, and then they differentiate. Yeah, but that's tentatively. Yeah, so that is by itself dynamic, no doubt. Yeah. Yeah. Yeah.
Yes, you including videos with slow rotation to be stimuli in the body. And we also that these regions, specific regions, more salient. I've seen the regions of the body that are often from ambient slower and fast. They are fibers, slow. And yet, these specific regions do other regions with after. So, how do you associate these calculations with the more anterior, which you see that regions slower to the body, which also have its own kind of things like this? Then,
Yeah, very good question. I mean, so we work a lot on breathing, because I got a student who was really interested in breathing. He was fanatic about breathing, so okay, so why not? Doing that, for me, it's just a time scale, nothing else. So okay, work with that. And we did a large-scale meta-analysis of all the breathing fMRI studies, whole brain, which of course is no surprise, because first it's a slow signal. That's what you also see in fMRI, and of course it's oxygen. I mean, it's a bold signal. I don't need to tell you this here at this place. Yeah, so of course, and you see the same for the heart studies. So I'm not an expert there in the heart, so you may know better. But I would say it's just they phase synchronized. They share mutual information. It's a cross-frequency talk. And I think the faster ones are integrated through probably maybe some top-down brain to body coupling. So because you have more power in the slower frequencies from the brain, they probably may enslave phase amplitude coupling the faster frequencies from the body, or the other way around also. So And there are in the breathing domain, there are a lot of cross-frequency coupling studies. You probably know them much better than I do because you're the insular specialist. How that now specifically relates to the insula, I refer to you. Yeah, but I think I would say there's even, I don't think actually that the brain would make a principal difference between external and environmental signal, because we can have, if you have the same frequency outside and inside, the brain probably processes them in the same way. Yeah. So it's a temporal distinction. That's why it's not it's not a principal distinction. It's a temporal distinction. That again, where you can learn a lot from psychiatric patients, because where you see these confusions. Yeah. So psychiatry can teach you a lot about them. Yeah.
Then one of the points raised for the questions really support any issue because science predictions.
Yeah. Okay, I see. Yeah, I mean, the question of faster and slower connection is yet a different question. I haven't addressed that yet. I mean, this would basically you go into dynamic functional connectivity and then calculate the dynamic functional connectivity in terms of its speed, and then